Imagine if every cell in your body suddenly stops dividing, your wounds would never heal, a tiny baby would never grow into an adult, even replacing the millions of cells that die every second would become impossible. Now imagine the opposite. What if cells divide continuously without any control? The result would be uncontrolled growth, leading to diseases like cancer. So the question is, how do our cells know exactly when to divide and when to stop and when to destroy themselves if something goes wrong? The answer lies in the most fascinating processes in biology, the cell cycle. Hello everyone, welcome back to Ciencia Vitae, where we'll explore biology through concepts that connect life. And in today's video, we'll take a complete journey through the cell cycle from cell growth to division, from mitosis and meiosis to cell regulation, checkpoints, apoptosis, and finally how failure of these mechanisms leads to cancer. So grab your notebooks and let's begin our journey into the fascinating world inside every living cell. So the question arises, what is cell cycle? So the cell cycle is the series of events that the cell goes through from one cell division to the next. It includes cell growth, DNA replication, and division into two daughter cells. So the cell cycle is generally divided into two major phases. The first one is interphase, which is the longest phase, and the second is M phase, which is the shortest phase. So first we'll study in detail about the interphase. Interphase consists of three stages: G1, S phase, and G2 phase. So G1 stands for gap 1. This is the first growth phase. During G1, the cell size increase and synthesis of RNA takes place. Proteins, organelles such as mitochondria and ribosomes. Essentially, the cell performs all of its normal metabolic activities while preparing for the DNA replication. At this stage, scientists often say that the cell asks itself, am I healthy enough to divide? If the answer is yes, the cell proceeds further, otherwise, it may pause or even enter a resting phase known as the G0 phase, where the cells remain metabolically active but do not divide. So the next phase is S phase, synthesis phase. This is one of the most important phases because DNA replication takes place here. Every chromosome duplicates itself. Remember, the chromosome number does not increase. Instead, each chromosome now consists of two identical sister chromatids joined together by a centromere. This distinction is extremely important for the competitive exam. The next is the G2 phase. G2 phase after DNA replication comes the gap 2 phase. Now the cell begins preparing for mitosis. It synthesizes proteins required from the spindle fiber formations. The centrosomes duplicate, more ATP is produced because cell division requires a lot of energy. And most importantly, the cell carefully checks whether DNA replication has occurred correctly or not. If any errors are detected, repair mechanisms becomes active before the cell enter mitosis. Now we'll study about the M phase, cell division to form two daughter cells. Although this is the shortest phase of cell cycle, it is the phase everyone remembers because actual cell division takes place here. The M phase consists of two processes. The first is mitosis, where the nuclear division takes place, and the second is cytokinesis, where the division of cytoplasm takes place. Together, these two produce two genetically identical daughter cells. So why is cell cycle important? What is the significance of cell cycle? Growth and development, repair and healing, replacement of old and damaged cells, asexual reproduction in unicellular organism. Note, if the cell cycle is not regulated properly, it can lead to uncontrolled cell division, which is cancer. The regulation of cell cycle. Cell division cannot happen randomly. Every cell division must be accurate. This is where proteins like cyclins and cyclin-dependent kinases come into action. Together, they regulate the progression of the cell cycle. In addition, specialized quality control points known as checkpoints ensure that damaged DNA is not passed on to daughter cells. We'll explore these checkpoints in detail later in the lecture. So we'll study about the differences between mitosis and meiosis. The first one is location. The mitosis takes place in the somatic body cells, whereas meiosis takes place in the germ sex cells in gonads, testes, and ovaries in both male and female. The purpose of mitosis is growth, repair, replacement, and asexual reproduction, whereas the purpose of meiosis is formation of gametes, reduce of chromosome number, and creation of variation. Number of division, one division takes place in meiosis, two division takes place meiosis 1 and meiosis 2. Number of daughter cells formed, two daughter cells are formed in mitosis, whereas four daughter cells are formed in the meiosis process. Chromosome number in daughter cells are same as the parent in mitosis, whereas it becomes haploid of the parent cell from 2n to n in the meiosis. Synapses of homologous chromosome does not take place in mitosis, whereas it takes place in the meiosis in the prophase 1 to form bivalent and tetrads. Crossing over does not take place in mitosis. It is present in prophase 1. What separates at the anaphase? Sister chromatids separate at the anaphase in mitosis, whereas homologous chromosomes separate in the anaphase 1, sister chromatids separate in anaphase 2. Genetic variation is not present in mitosis. Identical daughter cells are formed, whereas high genetic variation is present in the meiosis process due to crossing over and independent assortment. Result of mitosis, it maintains the chromosome number and genetic stability, whereas of meiosis, it reduces chromosome number and produces genetically diverse gametes. Moving on to next slide. We come to mitosis. So mitosis is the process of nuclear division in which one parent cell divides into two genetically identical daughter cells. It occurs in the somatic cells and maintains the chromosome number. It has six phases as you can see in the diagram. The first one is the interphase, also known as the preparatory phase for mitosis. In the interphase, cell grows and performs normal function. DNA replicates. Each chromosome consists of two sister chromatids. Organelles such as mitochondria increase in number, centrosomes duplicate and prepare for the spindle formation. ATP and protein required for cell division are synthesized. At the end of interphase, each chromosome consists of two identical sister chromatids. Joined together by centromere, the DNA content has doubled, but the chromosome number remains the same. Now we move on to prophase. Prophase marks the beginning of mitosis. Several important events occur simultaneously. The chromatin fibers condenses into short, thick, and clearly visible chromosome. Each chromosome consists of two sister chromatids attached by centromere. The red portion you can see between this blue, this is the centromere. The nucleolus gradually disappears. The nuclear envelope starts breaking down. Centrosomes migrate towards the opposite poles of the cell. Spindle fibers begin to form between the two poles. The spindle apparatus is essential because it will later ensure the equal distribution of chromosomes. Moving on to next phase, it is metaphase. Metaphase is characterized by the precise alignment of chromosome. During this stage, the chromosomes becomes maximally condensed. All chromosomes align at the center of the cell, forming the metaphase plate or the equatorial plane. Spindle fibers attach to the kinetochores located on the centromeres of the uh, chromosome. Each sister chromatid is attached to the spindle fibers originating from the opposite poles. Before the cell proceeds further, the spindle checkpoint verifies that every chromosome is correctly attached. This checkpoint prevents chromosomes missegregation. Moving on to next phase comes the anaphase. Anaphase is the shortest but the most dynamic stage of the mitosis. During this stage, the centromeres divide, the sister chromatids separate from one another. Each chromatid now becomes an independent daughter chromosome. Spindle fibers shorten, pulling chromosome towards the opposite poles. As you can see, the chromatids have separate and are moving towards the opposite poles. This equal movement ensures that both the daughter cells receive the same chromosome number. Moving on to next phase comes the telophase. Telophase is essentially the reverse of the prophase we have studied. The events include chromosomes reach opposite poles and chromosomes gradually decondense into chromatin. Nuclear membrane forms around each chromosome set. Nucleolus reappears and spindle fibers deassemble. At the end of telophase, two identical nuclei are present within the same cells. The last stage is cytokinesis. Cytokinesis is the division of the cytoplasm that follows nuclear division. In animal cells, a contractile ring is made up of actin and myosin forms beneath the plasma membrane, creating a cleavage furrow. The furrow deepens until the parent cell divides into two separate daughter cells. In plant cells, as plants, possess a rigid cell walls, they cannot form a cleavage furrow. Instead, Golgi vesicles accumulate at the center of the cell. These vesicles fuse to form cell plate. The cell plate develops into a new cell wall, creating two daughter cells. Importance of mitosis, growth and development of multicellular organisms, repair and replacement of worn out and damaged cells, asexual reproduction in some unicellular organisms, maintenance of genetic stability by producing identical cells. The spindle apparatus diagram, it is very important. It can come in university examinations and all. You can see centromere with the centrioles present here, spindle fibers arising, and chromosomes at the metaphase plate. Now we move on to meiosis I, which is also known as the reductional division, because it reduces the chromosome number from diploid 2n stage to haploid n stage. It occurs in the germ cells and produces genetic variation through crossing over and independent assortment. The stages of meiosis I, the first one is interphase, premeiotic S phase, where DNA replicates, each chromosome is duplicated to form two sister chromatids, chromosome number remains 2n. Then we move on to the prophase 1, longest and the most important stage. Homologous chromosomes pair synapses to form bivalents and tetrads. Crossing over occurs between two non-sister chromatids. Chiasmata formation, nuclear membrane and nucleolus disappear. Spindle fibers form. Now we move on to metaphase 1. Homologous paired bivalents align at the metaphase plate. Orientation of each pair is random. Independent assortment. Sister chromatids remain attached at the centromere. Moving on to anaphase 1, centromere of homologous chromosomes do not split. Homologous chromosomes separate and move to opposite poles. Sister chromatids remain together. Chromosome number is reduced from 2n to n. Telophase 1 and cytokinesis. Nuclear membrane may reform. Spindle fibers disappear. Cytoplasm divides. Two haploid N cells are formed. Each chromosome still has two sister chromatids. Result after meiosis I, two haploid N cells with duplicated chromosomes. Sister chromatids are still joined. Importance of meiosis I, formation of gametes, sperm and egg, maintenance of chromosome number across generations, creates genetic variation in population, important for evolution and adaptation. Note meiosis I is a reductional division, whereas meiosis II is equational division, which we'll study in the next slide. Now, meiosis II, equational division. Meiosis II is the second division of meiosis. It separates the sister chromatids similar to mitosis to produce four genetically different haploid cells. So before meiosis II, after meiosis I, two haploid N cells are formed. Each chromosome still has two sister chromatids. Moving on to prophase II, in each haploid cells, nuclear membrane breaks down. Spindle fibers form. Chromosomes still with two sister chromatids become visible. Moving on to metaphase 2, in each haploid cells, chromosomes align individually at the metaphase plate, which is the equator. As you can see, they have aligned. Chromosomes align individually and Then we move on to anaphase 2. In each haploid cell, centromeres split, sister chromatids separate and move towards the opposite poles. They have separated, as you can see in the diagram, and they're moving towards opposite poles. Telophase 2 and cytokinesis, nuclear membrane reforms, spindle fibers disappear, and cytoplasm divides. Two haploid end daughter cells are formed. Key points we can see in meiosis II. It occurs in haploid cells produced by meiosis I. Similar to mitosis in process, sister chromatids separate, chromosome number remains haploid, results in four genetically different haploid cells. Outcome is four genetically different haploid cells, and now we can move on to next slide. Cell cycle regulation and checkpoints. The cell cycle regulation ensures that the cell divide only when conditions are favorable and damaged DNA is not passed on to the daughter cells. This regulation is mainly controlled by the cyclins and cyclin dependent kinases, CDK inhibitors, and feedback loops. They work together to drive the cell through different phases of the cell cycle. If any error is detected, the cell cycle pauses to allow the DNA to repair, or if the damage is irreparable, the cell undergoes apoptosis. So, cell cycle checkpoints, there are three major checkpoints. The first one is G1 checkpoint, restriction point location at the end of G1. It determines whether the cell is large enough, has sufficient nutrients and growth factors, whether the DNA is undamaged. If all conditions are met, the cell enters the S phase. Otherwise, it pauses for repairs or enters the G0 phase. It monitors cell size, nutrient growth, and DNA integrity. Moving on to G2 phase, G2 checkpoint. Location, it is present at the end of G2 phase, checks if the DNA replication is complete and accurate, detects DNA damage. If okay, the cell proceeds to M phase. If not, repair is initiated or apoptosis is triggered. The next is M checkpoint, spindle assembly checkpoint, location, metaphase, anaphase transition. Checks if all the chromosomes are properly attached to the spindle fibers, ensure correct tension and alignment at the metaphase plate. If okay, anaphase begins. If not, prevent separation of sister chromatids. Now we move on to what happens if the checkpoints fail. DNA damage may be passed to daughter cells, chromosome missegregation may occur, can lead to mutations and controlled cell division and cancer. Outcome of proper regulation, accurate DNA replication, faithful chromosome segregation, healthy cell division maintains genomic stability. It is a summary table which you can use to revise in your exams. Moving on to apoptosis. Apoptosis is controlled cell death for a healthier tomorrow. It is a genetically programmed, controlled process of cell death that removes the damaged, old, and unnecessary cells without causing inflammation. It is essential for normal development, tissue homeostasis, and preventing disease such as cancer. Apoptosis occurs through two main pathways: intrinsic pathway, which is also known as the mitochondrial pathway, and the extrinsic pathway, also known as the death receptor pathway. In intrinsic pathway, it is triggered by the internal signals such as DNA damage or cellular stress. Mitochondria release cytochrome C, which activates the caspases, leading to the cell death. In extrinsic pathway, it is triggered when death ligands bind to cell surface, death receptors activating caspases and initiating the apoptosis. The activation capsase of intrinsic pathway is capsase 9, whereas in extrinsic pathway it is capsase 8. Executioner capsases are capsase 3, 6, and 7. So execution phase capsis activation takes place, the cleavage of substrates, DNA fragmentation, cell shrinkage and membrane webbing, formation of apoptotic bodies and phagocytosis. So, why is apoptosis important? Apoptosis is important for embryonic development, shape of organs and body structure, tissue homeostasis, removal of damaged and mutated cells, immune system, and infections, eliminate virus infected cells. Key regulators of apoptosis are the BCL2 family proteins, anti-apoptotic pro-survival proteins, pro-apoptotic Bax and Bac, BH3 only proteins, BID, BIM, Puma, Noxa, sense stress and activates Bax and Bac, inhibition of apoptosis proteins, IAPs, inhibit executioner gaspases and tumor suppressor. Very important gene P53 activates the pro-apoptotic genes Bax, Puma, and Noxa. Difference between apoptosis and necrosis. So the main difference is apoptosis. It is a programmed cell death, which is ATP independent and it is maintained throughout the life. It is it does not leads to inflammation and our outcome is apoptotic bodies, whereas necrosis it is uncontrolled cell death and it is not ATP dependent. The control is lost early and inflammation is present, random degradation and cell lysis takes place. Clinical relevance of apoptosis is present in cancer. Loss of apoptosis leads to cancer, autoimmune disease, reduced apoptotic deletion of self-reactive cells, neurodegenerative disorders, excessive apoptosis, therapeutic target drugs that modulate apoptosis, chemotherapy, BH3 mimetics. Remember, apoptosis is not the end but a necessary new beginning. Morphological changes which we can see in apoptosis. The normal cell undergoes cell shrinkage, chromatin condensation, membrane blibbing takes place, apoptotic bodies are formed, and phagocytosis takes place without causing any inflammation. Now we move on to cancer and cell cycle. Cancer is caused by the loss of normal cell cycle control, leading to uncontrolled cell division and tumor formation. The normal cell cycle control is that cell cycle is tightly regulated. Cyclins promote the cell cycle progression, CDKs drive the cycle forward, tumor suppressor like P53 stop cycle if something is wrong. Result leads to controlled division, normal growth, and tissue homeostasis. Whereas how cancer develops mutation in genes, inactive tumor suppressors, or activation of oncogenes, loss of checkpoints and cell cycle control, uncontrolled cell proliferation, tumor formation, oncogenes. Oncogenes mean gain of functions, drive the cell cycle forward, promote growth and proliferations. These are the examples. Tumor suppressor gene means loss of function, act as brakes on the cell cycle, prevents division when there is damage. Example P53, RB, APC, BRCA1/2 gene. Key players in cancer. Oncogenes are the accelerators, mutated overreactive genes that promote the cell growth. Tumor suppressor genes act as brakes, genes that stop the cycle or repair damage. DNA repair genes are guardian, fix the DNA damage and maintains the genomic stability. Checkpoints and cancer, G1 as checkpoint, G2 M checkpoint, and spindle checkpoint, which is present at the M checkpoint. This we have studied in the previous slides. The outcome leads to genomic instability, abnormal cells, and tumor formation if any of these checkpoints is not working properly. Outcomes of uncontrolled growth leads to sustained proliferation, invasion, and metastasis. Angiogenesis, new blood vessel formation, evading apoptosis, evading immune destruction. A summary of this slide, we can summarize it as cancer arises due to loss of normal cell cycle control. Oncogenes push the cell forward, tumor suppressor and checkpoints act as brake. Failure of repair and checkpoints leads to genomic instability. Understanding these helps in the targeted cancer therapies. So with this, I conclude the class on the cell cycle. I hope this presentation provided a clear understanding of mitosis, meiosis, cell cycle regulation, checkpoints, apoptosis, along with their importance in maintaining the normal cellular functions. Thank you all for your time and attention. I would be happy to answer any questions. You can drop them into comments or you can mail me. Thank you.